Okay, in one of our last videos, we discussed the structure and physical properties of carbohydrates, lipids, and protein. And now we're going to be talking about how these components are digested and how those components that are digested are absorbed into the body. So, of course, the location of this is going to be the small intestine. We've now passed from the oral cavity, ingested our dietary components, passed through the stomach, and now we're in the intestine. The importance of the small intestine is that this is a place where the pancreas can empty its enzyme content that it has synthesized into the small intestine. The pancreas is the organ that is producing the majority of the enzymes we use in digestion of dietary components. So let's start out by talking about carbohydrate digestion. Now, the process of digesting carbohydrates is a very simple biochemical mechanism. The enzymes are all part of a group called hydrolases. Hydrolases simply mean the enzyme is able to take water and break O-glycosidic linkages and take multimeric carbohydrates and break them down into their monomer structures. For example, we talked about starch. Starch is a glucose polymer. And the monomers of glucose are linked together in alpha 1,4 O-glycosidic linkages. And also recall we have branches on the polymers, and these are connected by 1 alpha 6 linkages. So in order to digest starch, to obtain the glucose monomers that we need for absorption into the body, we are going to use the enzyme amylase. This enzyme is a hydrolase, and it has the ability to take water and add it across the alpha 1,4 linkages. In doing so, this is able to hydrolyze off a glucose monomer off of the polymeric structure of starch. The next one we need to talk about, enzyme, is going to be processing the carbohydrate lactose. Lactose, recall, is a disaccharide, and it is composed of galactose and glucose. Now again, similar type of enzyme as amylase, except it has a differential substrate specificity, and this bond between lactose and glucose is broken by the enzyme called lactase. Once lactase performs this hydrolysis, it releases glucose and galactose as monomeric units that can be taken up again in the body. The final one we have to consider is our carbohydrate, dietary carbohydrate, sucrose. Recall, sucrose is a disaccharide of glucose and fructose. Similar nomenclature that we followed here with lactose, the enzyme sucrase performs the hydrolysis to cleave glucose and fructose releasing these monomers for uptake in the body. Now, once these components are released, we will now have the monomeric units that compose the majority of the carbohydrates in our diet. This being, of course, glucose, galactose, and fructose. These are all monomers. Recall the nomenclature that these are all hexoses. All three of these carbohydrates have 6 carbon backbone structures. Now, let's take a look at how these are absorbed into the body. What we have to consider is the cells that are involved in taking these monomers up are going to be lining the intestinal epithelium. So if this is the intestinal lumen at the top of the page, this would be the top of the cell that is lining the lumen. This layer of cells is called an epithelium. The epithelial cells are specialized cells that they have specific transporters to be able to take material up from the lumen and bring it inside the cell. Now, material has to be transported across this membrane at the top because adjacent cells are linked together by what are called tight junctions. These tight junctions regulate what is called pericellular transport. Pericellular transport is simply the movement of molecules between cells. Peri meaning around the cell. So tight junctions regulate this by controlling what goes through. And typically the only molecules that are small enough to get through these are ions and water. Larger components, like our dietary molecules, for example glucose, are far too large to fit through these tight junctions. So they have to be taken up by a process that is requiring the cells. Now recall that the membrane of a cell is made up of lipids. So this is not a compatible environment for the highly soluble, water-soluble structure of glucose. There's too many hydrogen bonding elements in glucose to keep it in the aqueous phase, and therefore it will not pass through the lipid organic environment. So therefore we need what is called a transporter. The transporter for glucose is a transmembrane protein. This transmembrane protein allows passage of material across this lipid layer because the protein itself can harbor an aqueous environment, such as a channel type activity. Its function as a transporter will bring hydrophilic molecules across the hydrophobic barrier and into the cell. For example, glucose can be bound specifically by this transporter and transported into the cytosol of the cell. Now this is a very, very important process because glucose is our primary carbohydrate energy source. So we want a process whereby we can make sure glucose is being transported in the cell. We're not going to just rely on simple diffusion control binding of glucose to the transporter. We are going to couple this with the co-transport of sodium ions. So this is a co-transport activity. What this means is that the transporter will only work when glucose is bound and a sodium ion is bound. Then both of these will be transported in simultaneously. Now, what powers all of this? Well, if we look at what is called the basal surface of the cell down here in this region, we will find another transporter. This transporter is effectively going to be transporting sodium out of the cytoplasm of the cell, and it will transport potassium inside the cell. Now, in order to do this, we need to make sure that there is enough energy, free energy, to drive this reaction. So you'll see me draw a little squiggle on this, and what this means is that this particular transporter is hydrolyzing ATP into ADP plus inorganic phosphate, and what we are getting out of this is energy. This energy is driving the transport of sodium out of the cell and potassium into the cell. Now notice these are moving in opposite directions, so this is called an antiporter. What this essentially does is use the energy of ATP hydrolysis to pump sodium out of the cell. This will establish a gradient of sodium, so that the gradient is high sodium on the outside of the cell and low sodium on the inside of the cell. This process is called primary active transport. Primary active transport is a transporter that uses energy, ATP, and acts as a pump. So we are literally pumping sodium out of the cell. Now what this is going to do is establish a gradient of sodium so that our sodium concentration is high in the lumen of the intestine, but recall again, it is low inside the cell. So by this gradient of a high concentration to low concentration in the cell, this low concentration being maintained by this primary pump, the sodium will drive this transporter's activity because we are following an osmotic gradient. We're moving toward a decreased sodium concentration. What this will do in turn is also drive the glucose transport inside the cell. This is called secondary active transport. It is secondary because it is operating because of the gradient established by the primary active transport. So a pump that is using a gradient established by a primary active transporter, use ATP, is always a secondary active transporter. Now once the glucose comes out of the cell, it has to enter the bloodstream, which is down here on the other side of the cell. In order to do that, we have a transporter that will take glucose and transport this out into the blood. This particular transporter is called GLUT2 for glucose transporter number 2. This does not require ATP. It also does not require a gradient established by a primary transporter, so it is not secondary active transport. This responds to the gradient of glucose glucose being higher in the cytoplasm of the cell than it is in the blood because it's being washed away in the blood, keeping the concentration low. So we're still following a gradient. This is called facilitated diffusion. Now, facilitated diffusion is simply allowing the molecule to flow through this transporter based on the gradient of itself alone and nothing else. Compare this to our secondary active transporter, which is called SGLT1. This stands for sodium, glucose, transporter 1. SGLT1 differs, of course, from the GLUT2 because it is a secondary active transporter and requires the co-transport of sodium. Now, let's go back here and address this sodium-potassium transporter. We see the sodium is driven out of the cell to establish this gradient, but what about potassium? Potassium could build up inside the cytoplasm so high that it could stop the activity of this transporter and want to intend to reverse because the concentration would be high inside and low outside. So the way we do that is to place a facilitated transporter for potassium in the cell, so this responds in the same manner as GLUT2, responds to the high concentration of potassium inside the cell and allows that to pass through. Once it passes through, it can then reverse itself on the sodium-potassium transporter. Now, if you recall the structure of glucose, you'll notice that it is very similar in structure to galactose. In fact, glucose and galactose are epimers. There's just one difference in the axial positioning of hydroxyl group. So therefore, because these two look so similar, galactose will follow the same path as glucose. So galactose in our diet will be taken up by the SGLT1 transporter. So instead of glucose, galactose can also bind here, and sodium can drive its passage inside the cell. Likewise, because it's so similar in structure, the GLUT2 transporter also recognizes galactose. And so the galactose, as well as glucose, can be taken through the GLUT2 transporter and transported into the blood. Now, what about fructose? Recall, fructose is another major monomer of our diet. Fructose simply passes through a transporter by facilitated diffusion. This transporter operates the same as GLUT2, except this transporter is specific for the carbohydrate fructose. This transporter is called GLUT5. I know, you want it to be called maybe FLUT5 for fructose transporter, but this is a misnomer because of purification back in the originally when these enzymes are identified as transporters. GLUT5 was thought to be a glucose transporter. It is now known to be a fructose transporter, but the name has remained. And likewise, in order to transport fructose out of the cell and into the blood, we need another transporter because this is a highly polar molecule. The transporter is also the same, GLUT5, on the basal surface. This operates strictly because of the concentration of fructose. There is no active transport. There is no primary active transport using ATP. There is no secondary active transport using a gradient established by ATP. The fructose simply has a high concentration in our lumen, and it will transport into the intestinal cell and then through into the blood by GLUT5. Now let's move on to dietary fats. So dietary fats, we saw, are largely contained in triacyl glycerol structures. Now recall, a triacyl glycerol is the glycerol backbone, and we have an ester linkage with our fats, our fatty acids, attached to each of the carbons of the glycerol backbone. Now, in order to get these materials across, these compounds across the cytoplasm and into the cytoplasm and into the blood, we need to break this up in the same manner as the carbohydrates. We need to hydrolyze these bonds. In this case, we are going to use an enzyme called lipase. Once again, this, like the other enzymes, are made by the pancreas and secreted into the intestine. The lipase is a hydrolytic enzyme, so therefore it's going to take water, and it is going to apply water to attack the carbonyl of the fatty acid attached to the triglyceride. It will do that 3 times. The end result is 1 glycerol molecule and 3 fatty acids. Now, alternatively, the lipase activity may be incomplete. And if it's incomplete, it will cleave the outside fatty acids from the glycerol backbone. For example, cleave number 1 and number 2 over here, but this one will not work, because lipases are highly specific for the fatty acids on the ends of the triacylglycerol structure. There's some activity, although be weak, to cleave the fatty acid on the middle carbon, number 2. The primary substrate is fatty acids on carbons 1 and 3. In this case, we will yield 2 fatty acids and 1 molecule called 2-monoglycerol. This is simply the glycerol backbone with a fatty acid remaining on the number 2 carbon. Now, in order to get these into the cell, we have to revisit our intestinal epithelial cell. And fatty acids, recall, have a long hydrophobic hydrocarbon chain and a charged carboxylic acid. So these long hydrocarbon chains, you would think, might allow it to pass through the lipid membrane, the organic environment. But it cannot, because of this carbohydrate, or this carboxylic acid residue. This is charged, so it will not pass this membrane. Therefore, we need a transporter. And this is called fatty acid transport protein, abbreviated FATP. FATP allows for the transport of the fatty acid into the cytoplasm. This occurs by facilitated diffusion. So again, this works strictly on the concentration gradient of fatty acids high in the lumen of the intestine and a lower concentration in the cytoplasm. Now, once this is in the cytoplasm of the cell, what we need to do is trap this inside the cell. Because recall, these transporters are reversible. We do not want this reverse activity. We want to maintain and keep a one-way flow. So the way we're going to do that is we're going to add a coenzyme A. We abbreviate it CoAsh for coenzyme A. This is a molecular component that we're going to be looking at a lot. It is conjugated using through this sulfur residue, and this is used to prepare molecules for next steps in biochemical reaction, and also to modify structures so that they can be participating in a one-way direction. So for example, we have an enzyme called fatty acyl-CoA synthetase. What this enzyme does is it takes the fatty acid transported by fat P, takes the coenzyme A, and couples these together. In this fashion. Now this structure is physically different than the fatty acid, so the fat P transporter will not recognize it. Now we have one more issue here, and that's the hydrophobicity of the hydrocarbon chain. This is not soluble in the cytoplasm. So how do we transport this in an aqueous environment of the transporter in the cytoplasm? What we need to do is call on another protein called fatty acid binding protein. The fatty acid binding protein, abbreviated FABP, structurally has been identified to look like a clamshell. So it looks like this. The hydrophobic portion of the fatty acid fits into a cleft of the enzyme. In doing so, it forms hydrophobic interactions with the interior of the enzyme, and that shields it from the aqueous environment of the cytoplasm. The coenzyme A moiety with the carbonyl can be pointed outward from the enzyme, and this will form hydrogen bonds with water and maintain solubility. So once this fatty acyl-CoA structure binds to the fatty acid binding protein, then this can be transported throughout the cell. Now the final structure we have to look at, of course, are proteins. Now we saw that proteins, their initial digestion occurred in the stomach, and they underwent an unfolding denaturation because of the low pH in the stomach, and then partial digestion occurred because of the enzyme pepsin. Recall that pepsin is from a general class of enzymes called proteases, enzymes that cleave proteins. Now we need to take a look at the different classes of proteases. Pepsin is one, but there are several others that that are made by the pancreas. And let's draw the backbone of a short protein. And we need to look at two different types of proteases. Some proteases will cleave the N-terminal amino acid by hydrolyzing the peptide bond that is holding the N-terminal amino acid onto the rest of the chain. This type of enzyme is called an aminopeptidase. Again, it's a hydrolytic enzyme. It adds water across this bond, but it's an aminopeptidase because it cleaves off the amino-terminal amino acid. Likewise, we have enzymes that can hydrolyze the peptide bond at the C-terminal amino acid. These are called carboxypeptidases. Their specificity is the amino acid at the C-terminal end. And finally, we have enzymes that will cleave peptide bonds within the linear primary structure. These are called endopeptidases. The endopeptidases recognize peptide bonds within linear sequences of amino acids. So from these three types of proteases made by the pancreas, secreted into the small intestine, we can have a complete digestion of our primary sequence of a protein into amino acids. This is a very efficient process because every time the endopeptidase cleaves, it releases another C-terminal and N-terminal amino acid that can be worked on by the amino and carboxypeptidases. Now the question is how do these proteases, how are they synthesized by the pancreas, secreted into the small intestine, and do not do any damage en route until they get to the small intestine? You can imagine if these enzymes were active in the pancreas, it would digest the pancreas. So what we have is the secretion of zymogens. Now we saw the zymogens. The first one we saw was pepsinogen, and pepsinogen is activated to pepsin by low pH in the stomach to become an active enzyme. In this case, what we have is the enzyme trypsinogen, which is a general class of endopeptidases. And trypsinogen is made by the pancreas in its inactive form. A series of other enzymes, aminopeptidases, endopeptidases, and carboxypeptidases, are also made and synthesized in the pancreas in their ogen inactive forms. When the trypsinogen reaches the small intestine upon secretion, what happens is it is converted into trypsin, which is active. This is done by an enzyme called enterokinase. The enterokinase is located on the epithelium of the small intestine. So this is a transmembrane protein on the luminal surface of the small intestine, and it activates the trypsinogen to trypsin. The trypsin now can mediate the activation of all the other proteases that are in the environment. So it's a cascade. Once all of these proteases have been activated, now we have the full complement of aminopeptidases, endo and carboxypeptidases, to work on the proteins in our diet. Now, the whole purpose of this is to take proteins and convert them into amino acids in the small intestine. Once we have this, then we can transport these molecules from the lumen of the intestine into the cell and into the blood. Now recall the structure of amino acids. These are generally polar or charged, and so these structures will also need a transporter. This transporter will bind amino acids, and it will transport them in conjunction with sodium. So, in a similar process as we saw with glucose uptake, this is secondary active transport. And also similar to glucose uptake, in order to generate this gradient, we have primary active transport, which is going to pump sodium out of the cell and pump potassium into the cell and hydrolyze ATP as the energy source to drive this gradient formation. Now there are facilitated transporters to take amino acids once they're in the cytoplasm and transport them into the blood. These, of course, work on the gradient established by the amino acid sodium transporter. The gradient is now used by the facilitated transporter to transport the amino acids out. One other note here is that the protein's digestion is not absolute. We can also have digestion down to di- or tripeptide structures. These di- and tripeptides have their own transporter to be taken up. Once these are inside the cell, these di- and tri-transporters are digested down to amino acids, and then they can feed into the stream of flow of amino acids into the blood. One final note is that the amino acid transporters have interesting characteristics to them. There is not a transporter for every one of the amino acids. The amino acid transporters can be considered class-specific. So what does that mean? We may have an anion transport for glutamate and aspartate, for example. We may have a cation transport for lysine and arginine. We may have a hydrophobic transporter, for example, for leucine and valine, etc. So amino acids are taken up primarily by the characteristics of their R chains. So their R chain will dictate the transporter that is being used.